Everything you have ever loved, feared, learned, or lost survives inside you only because memory keeps rebuilding it. Right now, somewhere behind your closed eyes, trillions of connections hold the shape of your entire life. The sound of a voice you haven't heard in years, the warmth of a room you once called home, the exact color of a sky on a morning that changed everything. None of it is written down. None of it is stored like a file on a shelf. It is alive, woven into the living tissue of your brain, reconstructed each time you reach for it. Tonight, as your breathing begins to slow and the weight of the day gently releases from your shoulders, we are going to travel inward, into the most extraordinary landscape you will ever know, your own memory. We will explore how the brain captures fleeting moments and transforms them into lasting impressions. How a single experience can etch itself into the architecture of your neurons, and how sleep, the very state you are drifting toward right now, plays an essential role in deciding what stays and what fades. Memory is not a recording device. It is something far more beautiful and far more fragile, a living process of construction, reconstruction, and gentle forgetting. Scientists have spent more than a century trying to understand where memories live, how they form, and why some persist for a lifetime while others dissolve like mist. The answers they have found are as humbling as they are wondrous. So let your body settle, let the quiet rise around you, and let us begin at the very place where every memory starts, with a single moment of experience. Consider, for a moment, the last time you stepped outside on a cold morning. Perhaps you remember the bite of the air on your cheeks, the way your breath rose in a pale cloud, the crunch of frost beneath your shoes. That single moment, ordinary as it seemed, ignited an astonishing cascade inside your brain. Light entered your eyes and was converted into electrical signals by roughly 120 million photoreceptor cells in each retina. Sound waves vibrated against your eardrums and were translated into nerve impulses. Temperature receptors in your skin fired rapidly, sending urgent messages along your spinal cord. The scent of cold earth drifted through your olfactory epithelium, where around 400 types of receptor neurons began sorting its molecular signature. All of this happened within milliseconds, and the raw information from every sense converged upon a small, ancient structure deep within each temporal lobe of your brain, the hippocampus. The hippocampus, named for its resemblance to a seahorse by the 16th century anatomist Julius Caesar Aranzi, is not where long-term memories are permanently stored. Rather, it serves as a kind of gateway, a temporary binding station where the scattered threads of an experience are woven together into a coherent whole. Without the hippocampus, new conscious memories cannot form at all. We know this in part because of a man known for decades in the scientific literature only as Patient HM, later identified as Henry Molaison. In 1953, at the age of 27, Molaison underwent an experimental surgery to treat severe epilepsy. The neurosurgeon William Beecher Scoville removed large portions of both his medial temporal lobes, including most of each hippocampus. The seizures diminished, but something else vanished too. From that day forward, Henry Molaison could no longer form new long-term memories of events in his life. He could remember his childhood, recall facts he had learned years before, and carry on a conversation, but minutes after meeting someone, he would forget the encounter entirely. His case, studied extensively by the neuropsychologist Brenda Milner and later by Suzanne Corkin, became one of the most important in the history of neuroscience, revealing that memory is not a single faculty, but a collection of distinct systems, each relying on different structures within the brain. To understand how a fleeting experience becomes a lasting memory, we must look at what happens at the smallest scale of the brain, at the junction between one neuron and another. The human brain contains approximately 86 billion neurons, a number established through careful counting methods by the Brazilian neuroscientist Susana Herculano Husel and published in 2009. Each neuron can form thousands of connections with other neurons, and it is at these connections, called synapses, that memory begins to take physical shape. In 1949, the Canadian psychologist Donald Hebb proposed an idea that would become one of the foundational principles of modern neuroscience. He suggested that when two neurons fire together repeatedly, the connection between them is strengthened. This principle, often paraphrased as neurons that fire together wire together, describes a process now known as synaptic plasticity. The specific mechanism that Hebb envisioned was discovered in 1973 by the Norwegian scientists Terje Lumo and Timothy Bliss, who were studying the hippocampus of a rabbit. They found that when they stimulated a neural pathway with a rapid burst of electrical pulses, the synapses along that pathway became stronger and remained stronger for hours, even days. They called this phenomenon long-term potentiation, or LTP. Long-term potentiation is now understood to be one of the key molecular processes by which memories are encoded. When you experience something meaningful, a conversation, a sunset, a moment of fear, specific networks of neurons fire together, and if the signal is strong enough or repeated enough times, the synapses between those neurons undergo chemical and structural changes. Receptor proteins called NMDA receptors on the receiving neuron detect that both the sending and receiving neurons are active at the same time, allowing calcium ions to flow in. This triggers a cascade of molecular events that ultimately makes the synapse more responsive to future signals. Over time, entirely new synaptic connections can sprout, physically reshaping the architecture of the brain. And so, a moment of experience, as weightless and invisible as a thought, leaves behind something tangible, a subtle but real alteration in the structure of living tissue. Your memories are not abstractions. They are inscribed, quite literally, in the geometry of your neurons. And yet, not all memories are created equal. Some moments burn themselves into your mind with startling clarity. A first kiss, a phone call bearing terrible news, the face of a stranger who showed you unexpected kindness, while other moments, equally real when they occurred, vanish without a trace. The difference, in large part, comes down to emotion. Nestled close to the hippocampus, almost touching it, lies another almond-shaped structure called the amygdala. The amygdala is central to the processing of emotions, particularly fear and arousal, and it has a profound influence on which experiences the brain chooses to preserve. When an event triggers a strong emotional response, the amygdala signals the adrenal glands to release stress hormones, including norepinephrine and cortisol. These hormones travel through the bloodstream and act upon the brain itself, enhancing the consolidation of memories in the hippocampus and across the cortex. Research by the neuroscientist James McGaugh and his colleagues at the University of California, Irvine, has demonstrated that moderate levels of emotional arousal strengthen memory formation, while extremely high levels can sometimes impair it or distort it. This is why traumatic memories can be both vivid and fragmentary at the same time, seared with sensory detail in certain places, yet strangely incomplete in others. There is a gentler side to this emotional encoding as well. Moments of warmth, love, comfort, and safety are also tagged by the brain as important. The neurotransmitter dopamine, released during experiences of pleasure and reward, plays a role in marking these memories for long-term storage. The feeling of a parent's hand on your forehead when you were ill, the laughter shared around a table with old friends, the quiet satisfaction of finishing something difficult. These experiences carry an emotional weight that tells the hippocampus to pay attention, to bind the details more tightly, to ensure that this particular pattern of neural activity persists. And so your memory is not a neutral archive. It is shaped by what you felt. The moments that moved you, whether toward joy or sorrow, wonder or fear, are the moments most likely to endure. Your emotional life and your memory are not separate systems. They are deeply, beautifully entangled, each one giving meaning to the other. Now let us turn to the very thing that is approaching you at this moment: sleep. For much of the 20th century, sleep was regarded by many scientists as a passive state, a kind of nightly shutdown during which the brain simply rested. We now know that this view was profoundly incomplete. Sleep is one of the most active and essential phases of memory processing. During the hours after you fall asleep, your brain replays the experiences of the day. This phenomenon has been observed directly in studies of the hippocampus. In a landmark series of experiments beginning in the 1990s, the neuroscientists Matthew Wilson and Bruce McNaughton at the University of Arizona recorded from neurons in the hippocampi of rats as the animals navigated a maze. Later, when the rats slept, the very same neurons fired again in the same sequence, as though the brain were rehearsing the journey it had taken while awake. This replay occurred primarily during a phase of sleep known as slow-wave sleep, also called deep sleep, which is characterized by large rolling electrical oscillations in the cortex with a frequency of roughly 0.5 Hz to 4 Hz. Scientists believe that during slow-wave sleep, memories that were initially encoded in the hippocampus are gradually transferred to the neocortex, the vast, wrinkled outer layer of the brain, for long-term storage. This transfer process is known as systems consolidation, and current evidence suggests it unfolds not in a single night, but over weeks, months, and sometimes years. The hippocampus acts as a temporary teacher, replaying patterns of activity and guiding the cortex to form its own stable representations. There is another stage of sleep that appears to play a complementary role: rapid eye movement sleep, or REM sleep, during which the brain becomes almost as electrically active as it is during wakefulness. Some researchers propose that REM sleep helps to integrate new memories with existing knowledge, finding connections and associations that were not obvious during the day. It is perhaps no coincidence that REM sleep is also when most vivid dreaming occurs. And so tonight, as you drift from wakefulness into the slow, deep rhythms of sleep, know that your brain is not shutting down. It is tending to your memories, sorting, strengthening, and weaving them into the story of who you are. As you drift closer to the edge of sleep, something remarkable begins to unfold within the architecture of your brain. During the deepest stages of non-rapid eye movement sleep, slow waves of electrical activity roll across the cortex like vast, unhurried tides sweeping across a darkened sea. These are known as slow oscillations, and they cycle at a frequency of roughly 1 Hz, one gentle pulse per second. Scientists have discovered that these slow waves do not merely accompany sleep. They actively orchestrate the transfer of memories from the hippocampus, where they were first encoded, to the neocortex, where they will be stored for the long term. It is as though the brain, in the quietest hours of the night, carefully sorts through the scattered notes of the day and files each one into its proper place. Research led by Jan Born and his colleagues at the University of Tübingen, published in the early 2000s, demonstrated that slow-wave sleep plays a critical role in the consolidation of declarative memories, the kind of memories that let you recall a face, a fact, or a moment in time. During these slow oscillations, bursts of faster activity are called sleep spindles. Brief, rhythmic pulses lasting 1 to 2 seconds appear to lock newly formed memories into the cortical networks where they will reside. These spindles, generated in the thalamus, act almost like a key turning in a lock, opening the cortex to receive what the hippocampus has been holding. And so the memory of a voice heard at breakfast, or a sentence read in a quiet room, is gently carried from its fragile temporary home into a more permanent one. You do not feel any of this happening. There is no sensation, no awareness. It occurs in perfect silence, beneath the surface of consciousness, while you rest in the deep stillness of dreamless sleep. The brain does not need your attention for this. It only needs your surrender, your willingness to let go, to stop reaching for the waking world, and to allow the slow tides to do their ancient, patient work. And yet, the brain's memory work during sleep does not end with slow waves. There is another stage, one filled with vivid, strange, and sometimes bewildering imagery that plays its own essential role. This is rapid eye movement sleep, the phase in which dreams are most often born. During REM sleep, the brain becomes intensely active, almost as active as it is during waking life, and yet the body lies still, held in a state of temporary paralysis, so that you do not act upon whatever scenes unfold behind your closed eyes. Current evidence suggests that REM sleep is especially important for the consolidation of procedural and emotional memories. If you spent the afternoon learning to play a new chord on a guitar, or if you navigated a difficult conversation that left you feeling unsettled. It is during REM sleep that those experiences are believed to be replayed, reorganized, and woven into the broader fabric of your understanding. The neuroscientist Matthew Walker, in his widely cited research, has described how REM sleep appears to strip the sharp emotional charge away from difficult memories while preserving their informational content. It is as though the brain, during dreaming, gently removes the thorn from the rose, allowing you to remember what happened without being wounded by it again each time the memory surfaces. Some researchers propose that this is why a painful experience often feels less raw after a night of sleep, and why the old advice to sleep on a problem carries a kind of quiet neurological wisdom. During REM sleep, the prefrontal cortex, the region responsible for logic and rational thought, becomes less active, while the amygdala and the hippocampus remain highly engaged. This unusual combination may be what gives dreams their surreal, emotionally charged quality. And it may also be what allows the brain to form unexpected connections between memories that seemed, during waking hours, entirely unrelated. Consider for a moment how gently this all unfolds. The dreaming mind, wandering through corridors of feeling, quietly healing and reorganizing without any conscious effort at all. There is something else worth holding in your mind as you settle deeper into the stillness. The idea that forgetting is not a failure. For a very long time, forgetting was understood as a flaw, a kind of erosion, as though memories were inscriptions in stone that simply wore away with time. But more recent research suggests that forgetting may be an active, purposeful process, as essential to the health of the brain as remembering itself. The neuroscientist Blake Richards, along with Paul Batt, published a paper in 2017 in the journal Neural, arguing that the goal of memory is not to preserve every detail of the past with perfect fidelity. Instead, they proposed that memory exists to help us make better decisions in the future. And for that, the brain must generalize, must let go of irrelevant specifics so that broader patterns can emerge. Imagine a path through a forest. If you remembered every single leaf, every insect, every shift of light, every occasion that you walked that path, the sheer weight of detail would make it difficult to recognize the path at all. By letting the irrelevant details fall away, the brain allows you to see the path clearly, to navigate it even when the leaves have changed and the light falls differently. This process of active forgetting appears to involve the weakening and pruning of synaptic connections, particularly during sleep. Some scientists believe that slow-wave sleep may serve a dual purpose, strengthening important memories while simultaneously allowing unneeded ones to fade. This is sometimes called the synaptic homeostasis hypothesis, proposed by Giulio Tononi and Chiara Chiarelli of the University of Wisconsin-Madison, which holds that sleep restores the brain's synaptic balance after a day of learning, has strengthened countless connections. And so, as you lie here, the brain is not only saving what matters, it is also, with great tenderness, releasing what does not. There is a quiet mercy in this. Not everything needs to be carried forward. Some things are meant to dissolve, like morning mist, leaving the landscape clearer and the mind lighter for the day ahead. Across millions of years of evolution, the ability to form and retrieve memories has been shaped by survival. Long before human beings developed language, long before the first story was ever told around a fire, memory already existed in simple forms. In the sea slug Aplysia, whose gill withdrawal reflex can be modified through experience, and in the honeybee, which can remember the color and scent of a flower once off the nectar. The Nobel Prize-winning neuroscientist Eric Kandel spent decades studying Aplysia, and through this humbling creature, he revealed some of the fundamental molecular mechanisms by which short-term memories are converted into long-term ones. He showed that short-term memory involves temporary changes in the strength of the synaptic connections, while long-term memory requires the synthesis of new proteins and the growth of entirely new synaptic structures. These findings, published over many years and recognized with the Nobel Prize in Physiology or Medicine in the year 2000, demonstrated that memory is not merely an abstract phenomenon. It is a physical transformation of the brain, written into the very shape and chemistry of its connections. And this is true of your brain as well, lying here in the quiet dark. Every lasting memory you hold has physically altered the structure of your neural tissue. The face of someone you love is not merely an idea floating somewhere in your mind. It is encoded in the pattern of synapses, in the particular arrangement of proteins, in the subtle geometry of connections between neurons. You carry the people and places and moments of your life not just in thought, but in matter, in the very substance of your brain. There is something profoundly intimate about this. The boundary between who you are and what you remember becomes almost impossible to find. Your memories are not possessions you keep in some separate room. They are the architecture itself, the walls, the windows, the light that falls through them. And tonight, as sleep deepens around you, that architecture is being quietly maintained, gently renewed, made ready for whatever tomorrow will bring. Consider for a moment the curious phenomenon known as a flashbulb memory. Those rare occasions when the mind seems to photograph an entire scene in extraordinary detail, preserving not only what happened, but the light in the room, the temperature of the air, the song that may have been playing somewhere in the background. The term was first introduced by psychologists Roger Brown and James Kulik in 1977, and their original proposal suggested that the brain possesses a special mechanism, triggered by surprise and emotional intensity, that captures moments with almost photographic precision. And yet, decades of research since then have revealed a more nuanced truth. Flashbulb memories feel exceptionally vivid and confident to the person who holds them. But studies, including a well-known investigation by Ulrich Neisser and Nicole Harsh following the 1986 Challenger space shuttle disaster, have shown that these memories are not necessarily more accurate than ordinary ones. People recalled where they were and what they felt with great certainty, and yet their accounts often shifted significantly over the span of just a few years, sometimes contradicting what they themselves had written down shortly after the event. What makes flashbulb memories so compelling, it seems, is not superior recording, but superior emotional tagging. The amygdala, that almond-shaped structure nestled deep within each temporal lobe, responds powerfully to moments of high emotion. And when it does, it signals the hippocampus and surrounding cortical areas to strengthen the encoding process. The result is a memory that feels crystalline, that carries with it the warmth and weight of the original moment, even if certain details have quietly drifted from their true positions over the years. This is a beautiful imperfection, one that tells us something profound about what the brain considers worth saving. It does not prioritize perfect accuracy above all else. Instead, it prioritizes emotional meaning, the felt significance of an experience, the way a moment mattered to the person living through it. And so, as you breathe slowly now, you might notice how even your own most vivid memories carry not just images, but feelings, textures of emotion draped gently across the surfaces of the past. Somewhere in the quieter reaches of neuroscience, there exists a question that still draws wonder from those who study it. The question of how a memory, which begins as a fragile and temporary electrical pattern in the hippocampus, gradually transforms into something far more stable and distributed across the vast landscapes of the cerebral cortex. This process is known as systems consolidation, and current evidence suggests it unfolds not over minutes or hours, but over weeks, months, and sometimes years. In the early stages of a memory's life, the hippocampus acts as a kind of temporary index, binding together the scattered cortical regions that each hold a different fragment of experience. A colour stored here, a sound stored there, a spatial context held in yet another area. Each time the memory is reactive, whether during waking thought or during the quiet replays of sleep, these cortical connections are believed to strengthen incrementally, until eventually the memory can be retrieved without the hippocampus serving as the intermediary at all. Some researchers describe this as the memory slowly graduating from hippocampal dependence to cortical independence, a process elegantly demonstrated in studies of patients with hippocampal damage, who often lose recent memories while retaining those formed many years before. This pattern, first described in the context of a patient known as Henry Mollison, referred to in the literature for decades as HM, provided one of the most important clues in the history of memory science. After portions of his medial temporal lobes were surgically removed in 1953 to treat severe epilepsy, Henry could no longer form new lasting memories, and yet his childhood recollections remained largely intact. His case, studied extensively by the neuropsychologist Brenda Milner, and later by Suzanne Corkin, revealed that the hippocampus is essential for creating new declarative memories, but that older memories had already migrated elsewhere. And so the architecture of remembering reveals itself as a living process, a slow migration of meaning from one region to another, the brain gently redistributing its treasures across the cortex, like sediment settling across a wide and ancient river. There is another kind of memory that moves through you even now, though you may not be aware of it. The kind that lives not in conscious recall, but in the body itself, in the effortless coordination of muscles and movement and breath. This is procedural memory, sometimes called implicit memory, and it is the reason you can ride a bicycle decades after last touching one. The reason a pianist's fingers can find a melody before the conscious mind has named the first note. Procedural memories are encoded through a different set of structures than declarative ones, relying heavily on the basal ganglia, a collection of nuclei nestled deep within the brain, as well as the cerebellum, that densely folded structure at the base of the skull that contains roughly 80% of the brain's total neurons, despite being only about 10% of its volume. These regions work together to store the patterns of skilled movement, refining them through repetition until they become automatic, until the conscious mind can step back and allow the body to remember on its own. This distinction between conscious and unconscious memory was powerfully illustrated by cases in which patients with severe amnesia, patients who could not remember meeting someone 5 minutes earlier, could nonetheless learn new motor skills and show improvement from day to day, even though they had no memory of having practiced at all. The knowledge was there, held in a system that did not require awareness to function. And perhaps there is something deeply comforting in this, in knowing that so much of what you have learned, how to walk, how to speak, how to hold a cup, how to breathe in just the way you are breathing now, lives within you as a quiet and enduring presence, independent of any effort to remember. These memories do not need to be recalled. They simply are, woven into the fabric of your movements, present in every gesture, steady as a heartbeat. Let that steadiness carry you now, as the body remembers what the mind no longer needs to hold. And yet, for all the elegance of the brain's memory systems, there is one quality of remembering that remains among the most mysterious and perhaps the most beautiful. The way a single sensory detail can unlock an entire world that you thought was lost. The scent of rain on warm pavement, the particular golden light of a late afternoon in autumn, a few notes of a half-forgotten song drifting from an open window, and suddenly you are somewhere else entirely, immersed in a moment you had not consciously thought about in years. This phenomenon, sometimes called involuntary autobiographical memory, was described with extraordinary literary precision by Marcel Proust in his novel In Search of Lost Time, published beginning in 1913, in which the taste of a small cake dipped in tea awakens an entire cathedral of childhood memory. Neuroscience has since explored the mechanisms behind such experiences, and current evidence suggests that sensory cues, particularly those involving smell, have a uniquely powerful capacity to trigger autobiographical recall, because the olfactory system projects directly to the hippocampus and the amygdala, without first passing through the thalamus, the relay station that most other senses must traverse. This direct anatomical pathway may explain why scent-triggered memories tend to feel more emotional and more immersive than those triggered by sight or sound alone. Some researchers have described this as the Proustian memory effect, and studies using functional magnetic resonance imaging have shown that odor-evoked memories are associated with stronger activation in the amygdala and hippocampus compared to memories triggered by other sensory modalities. And so the architecture of the nose and the brain conspire to preserve something tender, something that facts and dates alone could never capture. The felt quality of a moment, the atmosphere of a place, the way it felt to be alive in a particular room at a particular hour. Let yourself drift now into whatever gentle memory rises of its own accord, knowing that the brain has kept it safe, tucked away in the folds of sensation, waiting for exactly this quiet moment to return it to you. Consider for a moment the curious phenomenon of forgetting, because forgetting is not a failure of memory, but one of its most essential functions. Every second of your waking life, your senses deliver roughly 11 million bits of information to your brain, and yet your conscious mind processes only a tiny fraction of that torrent, perhaps 40 to 50 bits per second. If every detail of every moment were preserved with perfect fidelity, the precise angle of light through a window at 2:15 on a Tuesday afternoon, the number of ceiling tiles in a waiting room, the faint hum of a refrigerator 3 rooms away, the result would not be wisdom, but paralysis. There is, in fact, a documented case that illustrates this with haunting clarity. Solomon Cheryshev, a Russian journalist studied extensively by the neuropsychologist Alexander Luria in the early 20th century, possessed a memory so extraordinary that he could recall lists of words, numbers, and nonsense decades after hearing them only once. His mind was a palace of near perfect retention. And yet Cheryshev struggled profoundly with abstract thinking, with recognizing faces that changed expression, with understanding metaphor and poetry. He could not forget, and so the world became an overwhelming cascade of detail without hierarchy, without meaning. His story suggests something profound, that the architecture of memory is not designed merely to hold, but to select, to weigh, to let certain things dissolve so that others may stand in sharp relief. The neurobiologist Yadin Dudai and others proposed that active forgetting, sometimes called adaptive forgetting, is a deliberate biological process, not simply the passive decay of unused connections. Certain mechanisms appear to prune synaptic connections, clearing space and reducing interference, so that the memories most relevant to your survival, your identity, and your emotional life may be retrieved more efficiently. And so tonight, as the small events of this day begin to soften on the edges, as details blur and recede like stones beneath a rising tide, know that your brain is not losing anything of importance. It is sculpting. It is choosing what to carry forward into tomorrow. And there is one more quiet miracle worth resting with before sleep claims you fully. The way memory binds itself to place. Neuroscientists discovered, through decades of careful work, that a small seahorse-shaped structure, called the hippocampus, the very region so central to forming new memories, is also deeply involved in spatial navigation. In 1971, the neuroscientist John O'Keefe recorded from neurons in the hippocampi of freely moving rats, and found something remarkable. Certain cells fired only when the animal occupied a specific location in its environment. He called them place cells, and their discovery, which contributed to his Nobel Prize in Physiology or Medicine in 2014, revealed that the brain constructs an internal map of the world, and that this map is woven from the same neural fabric as memory itself. This is why, when you walk into a room you have not visited in years, memories may surface unbidden. Not as deliberate recall, but as a sudden, almost physical sensation of the past folding into the present. The smell of old wood, the quality of light against a familiar wall, and suddenly you are not merely remembering a place, You are remembering who you were when you last stood there. Place and self become inseparable threads in the same tapestry. Later research by Maybrit Moser and Edvard Moser identified grid cells in the entorhinal cortex, neurons that fire in precise hexagonal patterns as an animal moves through space, creating a kind of internal coordinate system. Together, place cells and grid cells form a navigation system of astonishing elegance. And this system is not separate from memory, it is its foundation. Some researchers now believe that episodic memory, your ability to mentally travel back through time and relive a specific moment, may have evolved from these ancient spatial circuits, repurposed over millions of years from mapping physical terrain to mapping the terrain of experience. So perhaps every memory you hold is in some deep sense a place you once stood. And tonight, as you drift, you are simply traveling gently from one remembered landscape to another, each one softened by distance, each one still quietly yours. And so here you are, at the quiet edge of sleep, carrying within you an archive more intricate and more precious than any library ever built. Every person you have loved has left a trace in your synapses. Every place you have called home still lives in the firing patterns of your neurons. Every lesson you have learned, every kindness you have received, every morning you have watched the light return, all of it persists, held gently in the living tissue of your brain. Tonight, as your breathing deepens and the world outside grows still, your mind will begin its quiet work, replaying, sorting, strengthening the threads of your experience. You do not need to do anything. You do not need to hold on. Sleep will carry you, and your memories will take care of themselves, the way they always have, the way they always will. Let go now. You are safe, and everything that matters is already saved. If this journey into the science of memory brought you a sense of wonder and calm, you might consider subscribing so that more evenings like this one can find their way to you. Leave a quiet thought in the comments if you wish. Perhaps a memory you are grateful the brain chose to keep. And when you